How one can define reportable range for impurity during conducting method validation? Because range is one of the essential parameter for validation. In this video, we will try to understanding the reporting ranges for impurity method validation according to the ICH guideline Q2R2. Hi, my name is Bhaskar Napte. I am the founder of Pharma Growth Hub and I am on the mission to provide an absolute clarity on such a very important technical topics to the pharmaceutical professionals. In case if you want to join our community, you will find the details in the description below. So let us begin with the discussion now. And I already made videos on understanding the reportable ranges for assay and dissolution. So you will find the details into the description below as well for those videos. So here is the first important point, understanding the reportable ranges for impurity or related substances. So there is a one overshare and we will talk about this point number 1 at the end of this video. Let us begin our talk by understanding how to define the low-end reportable range. See, the range always has the different concentrations starting from low concentration to the high concentration. So this particular column talks about understanding the lower concentration or the lowest concentration for beginning of our range. So what is the guidance provided by ICH guideline Q2R2? And the guidance says that you can consider a reporting threshold to define your low end of the reportable range study. So where you will find the reference for a reportable threshold? See, the reportable threshold can be understood by two different ways. The first one is, and let us understand the definition of reportable threshold. Now this is the concentration or the content of impurity, it could be known, unknown, needs to be reported. And when the impurity can be reported, by the way, the impurity cannot be reported unless and until it is quantified precisely and accurately. Now what is that concentration that is going to help us to report the impurities precisely and accurately? Actually, I am taking you to a very important concept. So what is that concentration? What is that concentration which can be reported precisely and accurately? I'm talking about the definition part of one of the very important parameters, which is also needs to be performed during method validation. Needs to be reported accurately and precisely. That is nothing but limit of quantitation or quantitation limit. Means our reportable range could be just equal to quantitation limit, isn't it? Our reporting range or reporting the reporting threshold can be our quantitation limit. But again, there is a requirement. If I have the method in the place, can I have the reporting quantitation limit as 0.1% or 0.2%? No. We need to define what is the requirement given under the ICH guideline Q3A and Q3B for the reporting thresholds. And we'll talk about those threshold details in the next slide. But I hope you are clear on the first point, that is talking about how one can correlate quantitation limit with the low end of the reportable range. So if my quantitation limit is 0.02%, that becomes my low end. If my quantitation limit is 0.04%, then that becomes my low end of the reportable range. Let us now understand what is the expectation defined for reporting threshold by the ICH guideline. And there are two important guidelines needs to be understood. The first one is Q3AR2. Now this talks about impurities in new drug substances, and Q3B talks about impurities in new drug product. So what is the reporting threshold requirement for the new drug substances according to the Q3AR2? And you will find the details over here. And there are two important information needs to be understood. If the maximum daily dose is less than or equal to 2 grams per day, then our reporting threshold should be 0.05%. If the maximum daily dose is greater than 2 grams per day, then the reporting threshold will be 0.03%. Now here is the important information. So we connected the low end with the limit of quantitation or quantitation limit in the previous slide. Now let us understand, if my reporting threshold, let us assume that we are working on to the drug substances method validation, and the maximum daily dose is less than 2 grams per day. So 0.05% is going to be our reporting threshold, right? Our reporting threshold is going to be 0.05%. I must confirm that my limit of quantitation is not more than 0.05%. Means if I have the quantitation limit equal to 0.01% or 0.03% or until 0.05%, it is okay. It is acceptable. But in case if the quantitation limit is, let us say, 0.1%, so it won't be acceptable. It won't be accepted because it is more than our reporting threshold. And understand, we're talking about the reporting threshold of 0.05%. In that case, my quantitation limit maximum can only be 0.05%, but should not be more than 0.05%. So the low end of my range study should begin with my quantitation limit to be very precise and accurate with the requirement. So you have to define the quantitation limit, understand what is the concentration required, and from that concentration onward, you can plot your range. You can define the linearity at that particular, starting from that particular concentration until your highest end. We'll talk about the high end very soon. You can understand whether your method is precise, accurate, and specific at this low end concentration. Now, how to understand this 0.05%? Let us assume that your quantitation limit, let us assume that the quantitation limit is 0.05%. So how to understand the concentration in terms of microgram per ml? Always remember, your 100% concentration is equal to your sample concentration. So what is your sample concentration? You say my sample concentration is maybe 1000 ppm. So 1000 ppm becomes 100%. Will you be able to now calculate 0.05%? So x ppm is going to be 0.05%. So how much is x ppm now? It is going to be how much? Is it 0.5 ppm? So it is going to be a 0.5 ppm, isn't it? So 0.5 ppm or 0.5 microgram per ml is the concentration you need to select for your low end range. I hope you understand this point as well. And the same is the case if you want to understand the requirement for drug product. The reporting threshold values are on the screen, and it is again dependent on to the maximum daily dose. If the maximum daily dose is maybe less than or equal to 1 grams per day, then 0.1% is your reporting threshold. But in case if the maximum daily dose is greater than 1 grams per day, then the 0.05% is your reporting threshold. So I hope you understand defining the reporting threshold concentration, right? We talked about this example, 0.5 ppm with respect to 0.05%. And I hope you are able to connect now the requirement of quantitation limit, reporting threshold with the low end of your range study. Let us now understand the high end in the next slide, how to define the high end concentration. So high end concentration can be defined by understanding this statement. 120% of specification acceptance criteria, and here is the additional information given, which is not part of the guideline by the way, but you need to consider this point also. Always consider shelf life spec concentration. See, in most of the times, you will have the two different specification. One is release spec and another is maybe the shelf life spec, shelf life specification. So what is, let us say you have impurity A, and for impurity A, your release spec is maybe how much? Let us assume that your release spec is 0.5%, not more than 0.5%, and your shelf life spec is 1.0%. So what is going to be now your high-end reportable range? Is it with respect to 0.5% or is it with respect to 1%? You need to always calculate this 120% with respect to shelf life specification. Why? Because this is going to be the worst case concentration. Just understand, if I calculate 120% of each specification, now this will become how much? 0.6%, isn't it? I'm just calculating 120% of working concentration at the release peak, and if I calculate the 120% of the shelf life concentration, that will become 1.2%. So if I actually measure the range until 1.2%, I am also including this 0.6% concentration also. But if I just measure the range with the high points at 0.6%, I will not understand whether my method is precise, accurate, or linear at 1.2% concentration. So it is very important to realize that this 120% specification acceptance criteria should be from the shelf life specification, because shelf life spec is always going to be, if not equal to the release spec, but can be the higher than the release spec. But in case if you have the same specification for shelf life and release, then no worries, you can consider any specification. Now how to understand this 1.2% now? Understand the sample concentration. Let us say sample concentration is 1000 ppm. So 1000 ppm is your 100%, and then calculate this how much? Let us say our 1.2% is going to be how much ppm. So how much it is going to be? Is it going to be a how much? 12 ppm if I'm not wrong. 12 ppm, right? 12 ppm, right? So what is your range now? Your low end concentration is, if you remember our earlier talk, 0.5 ppm or microgram per ml, and our high end concentration is 12 ppm. And then you can define 5 or 6 levels in between these two concentrations. Then you need to prove that our method is precise at 0.5 ppm, at 12 ppm also. You need to also confirm that your method is accurate at 0.5 ppm and at 12 ppm also. So this is the importance of defining the range for different types of test procedures. I hope you are clear on to the defining the low end and high end reportable range for an impurity. Let us understand about the purity part now. So some methods are always calculate the impurities based on to the area percent. Area percent means what? You are going to calculate the area from all the impurities plus your principal compound or drug substance, and then the percent area will be calculated for each and every impurity. That is called as the percent area basis, as percent area. In case of this point, impurity in the first discussion, you may be calculating the impurities against the diluted standard, against the external standard. Now, what is the need of having this external standard? I have video made onto this particular topic. So please search on this topic and you will get the video in when you can use the external standard method and when you can use the percent area basis or the area normalization calculation for impurities. So in case if you are using the area normalization or percent area calculation, your low end is going to be 80% of your lower specification acceptance criteria. So we talked about shelf life and the release specification. And most of the times our release specification can be a little stringent, little lower. If you remember, we talked about 0.5% as a release spec and 1% as a shelf life spec for impurity A. So always take 80% of now the release specification. So what is the specification we have release spec? Let us say we have release spec is not more than 0.5%. So 80% of that, 80% of zero point 0.5 is going to become how much? Is it 0.4%? 0.4%. Now, what is your sample concentration? Use the same logic. My sample concentration is 1000 ppm, and that is going to become my 100%. So, how much is going to be my 0.4%? This 0.4% is going to become how much? 0.4% is going to become how much? Is it going to become 40 ppm, right? So it is going to become 40 ppm. I hope you understand how I calculated the concentration for the low end, that is the 40 ppm, with this very simple calculation formula. Or is it a 4 ppm if I'm not wrong? 0.4% is going to become how much? Let me do it quickly. Why to unnecessarily give wrong values? So here I go with the calculation now. You can also do along with me 1000 into zero point 0.4 divided by 100, and that gives 4 ppm. Okay, I'm so sorry for that. It gives 4 ppm. 0.4 is percent is 4 ppm. Clear? Let us now understand the high end concentration in case of the area percent method, and upper specification acceptable criteria or 100%. So what is your upper acceptance criteria or 100%? That itself is going to suffice the requirement of the, if the impurities are calculated on as is basis or on the percent area basis. So let us say we have the specification that talks about release spec is not more than 0.5% and the shelf life spec is not more than 1%. Not more than 1%. So what is your high end concentration going to be? Not more than 1%. Means upper specification is not more than 1%. So that is going to define our high end concentration in case we are using or calculating the purity, or we are calculating the impurities on percent area basis. We talked about the another important point, and that is here now. This is the point number 1. So what is the point 1 denotes? Now where assay and impurity are performed as a single test and only one standard is used, linearity should be demonstrated for both the reporting level of the impurities and up to 120% of the specification acceptance criteria for assay. Now this defines the requirement for assay. See, most of the times, in case if I'm using the purity as a measurement for impurities against area percent, I may have the same standard for I may have the same standard concentration and the sample concentration. What does this means? Let me explain with the help of example. Let us say my standard concentration is 1000 ppm and my test concentration is also 1000 ppm, isn't it? So in this case, especially for drug substance, I need to draw a linearity. I need to draw a linearity from my low end concentration is what now? The 80%, not 80%, but here for impurity, it is reporting threshold. Let us say from quantitation limit to 120% of this 1000 ppm. This is the requirement mentioned under the point number 1. So I need to define the range linearity from quantitation limit to 120% of the sample concentration, and this is only applicable for the drug substance. This need not to be studied for impurities. That has been discussed. This is only for unknown impurities. This is only for the linearity to be studied for the drug substance. Okay, so quantitation limit could be, maybe we talked about how much, 0.5 ppm, right? 0.5 ppm. So this is my new range, 0.5 ppm to 120% of 1000 ppm, and this becomes how much ppm, by the way? 1200 ppm. This becomes 1200 ppm. That is 120% of 1000 ppm, and this is only applicable for the API, right? This is only applicable for the linearity. This is only applicable for the linearity. I hope you are clear with this important additional point. What is mean by the point number 1? So please let me know in case if you have any question for me in the comment box below, and I will be happy to answer all your questions. Thank you so much.